Hello, good afternoon. We are going to be starting lecture 3 today, and at the end, I'll take up those poll questions that we didn't have a chance to do for lecture 2 from last week, and hopefully that will help prepare you for the upcoming quiz, which is on Thursday. So lecture 3 is about cells. We have learned um, about atoms and molecules from lecture 2 and macromolecules as well. So now we're kind of moving up this hierarchy of organization. And we'll be learning about cells for quite a bit, all the way up to lecture 9. And then in lecture 10, we will learn tissue. And then that will set you up with a nice transition to the next semester, where you'll be spending most of the time learning about organs and organ system. So cells are pretty small. This gives you like a relative scale. A chicken egg is about between 10 millimeter and 100 millimeter. 100 millimeter is roughly I guess that would be like for the ostrich egg, right? But like a standard chicken egg, it's about 2 to 4 centimeter wide, tall. And then if you go down the scale couple times, then you can see animal and plant cells, they are measured in the range of micrometers, okay? So micrometer is this symbol here. I showed you this before. 1 meter has 1 million micrometer. So these are too small to see with the naked eye, and we need to use a microscope to help us see it better. Organelles are even smaller. They are found inside these animal cells or plant cells, and they have various function, and they are as small as 1 micrometer compared to, 50 micrometer, for example, for the entire cell. They are a similar size as bacteria. Viruses are even smaller. They're measured in nanometers. 1 meter has 1 billion nanometers in them. Okay, so this just gives you a little bit of an idea of how big these things are. So what is a cell exactly? By definition, cell is defined as the smallest unit that has the properties of life. All the characteristic we talked about in lecture 1, the ability to maintain homeostasis, complexity, reproduction, right? All these things are still true, are still retained by a cell. If you go any further into the cell, if you look at the organelles, then some of these properties are no longer valid. Right? So the smallest unit that retains the characteristics of life is fundamentally defined as a cell. Now, cells comes in different shapes and different forms. For example, over here, we have some bacteria, and there are different kinds of bacteria. These are little balls, so we call them cocci. And over here on the top right, we have something called an amoeba. You'll find amoeba in like pond waters. Okay? Sometimes amoeba might find themselves into like a swimming pool or something, and it could cause infection for people who ingested them. Okay? Anybody know what this might be? The picture at the bottom right.
Nerve?
That's right. These are neurons, right? So we have neurons in our brain, in our spinal cord, and pretty much throughout the body. Um, I mean, like you can feel things with your if something is brushing against your hand, you can feel that, right? That signal is transmitted by nerves, right? So they're kind of all over the place, and that's what they look like, right? So even though cells look very different, depending on what organism or what part of the body you're looking at, they all tend to share a set of features. All cells, regardless of type and size and source, they have a cell membrane. Right? So you can think of the cell membrane as the barrier, the boundaries of a cell. Okay, outside of the cell membrane would be its we call it the extracellular environment, EC ECF, extracellular fluid. I can write that out for you. Extracellular fluid. And then the inside would be what we call the cytoplasm. Okay? And then this boundary is the cell membrane. Okay? So cell membrane sometimes is also called a plasma membrane. And inside the cell is this like almost like gel-like substance that's made up of primarily water, and we call that the cytoplasm. Okay. Now there's also some kind of genetic material. Okay, I'm just gonna put the genetic material here. Okay, so that's the genetic material. Sometimes the genetic material is just floating around on its own. Other times it's packaged nicely in a structure. And we'll talk about the difference in a bit. But generally, these are the three things that are always going to be there for any cell. Okay, cell membrane, genetic material, cytoplasm. Let's talk about the cell theory. The cell theory has 4 parts to it. 4 generalization makes up the cell theory. First statement, every living organism consists of 1 or more cells. If something has a single cell, then we call them unicellular. Okay. On the other hand, if it has more than 1 cells, we call them multicellular. So here we have something called a paramecium. It's related to the amoeba. They're the same category of organisms. And both the amoeba, in fact, and the paramecium, they both live in fresh water. And they are just 1 cell. Okay? The entire organism is made up of a single cell. Okay? So that's unicellular. Here we have something called a C. elegans. C. elegans is a model organism. Okay? Model organism. A model organism is an organism that scientists knows a lot about. They know about all their genes, they know about the growth cycle and everything, and they are used for research. Okay, if people want to test like a new drugs or something like that, a lot of times before going all the way to humans, they start with these model organisms. Okay, so this one, it's almost like a worm-looking organism, and it has about 3,000 cells in it. And then humans, there are a lot of cells in us. Okay, trillions of cells. Like obviously, when you grow up, you would have more cells than you did when you were a baby. But compared to some of these other organisms, we have like way more cells than them. Okay, so that's the first part of the cell theory. Every living organism is going to have 1 or more cells. If it's living, it's going to have cells. And if it has cells, it's living. Part 2. A cell is the smallest unit of life. We already briefly talked about that. They have the properties of life, but more importantly, they are individually alive, even as part of a multicellular organisms. All the cells in your body has life on its own. You can actually take out most of the cells from the body, and they would be able to grow in these are called petri dish. They will be able to grow in a dish for some time. If you supply them with nutrients, if you give them the right condition, the right temperature, they will be able to survive outside of the body, okay? Because they are individually alive. Okay? Now there was this story about a woman named Henrietta Lacks. Her name is not on the page, but yeah, that's her name. Okay, Henrietta Lacks. And what happened was she had cancer, cervical cancer, and the people who were treating her at the time took a sample, took a biopsy of her cancer cell, and they tried to grow it in a dish. Okay? And they found out to their surprise, they were able to keep on growing these cancer cells for a very, very long time. They keep on dividing endlessly as long as you give them nutrients. Okay, like if they get too crowded, you can put them in a new plate, and they would just keep going. Normal cells don't do that. Yeah. So like if I take your cells, your healthy cells, and I grow it in a dish, they would be able to survive for some time. But even under optimal condition, they would eventually stop dividing and die. Okay, but not for the cancer cells. Cancer cells can grow virtually forever as long as you feed them. Okay, and so that created what comes to be a cell line. Right? So there are many different kinds of cell lines that you can buy nowadays from companies, and they are basically immortal cell lines that you can just purchase and grow in your own lab, and you can do experiments on them. And so that's what the HeLa cells look like. It's named after her, right? Henrietta Lacks. And these HeLa cells are actually being used for early studies for many new medicines. And a lot of the treatment for various diseases like TB, Ebola, HIV, and even cancer treatment, they all began testing using these HeLa cells as the first step. Okay, we call it like the proof of principle. You have to show that it actually works in a dish first before you can move up to animals like mice, and then you can maybe move up to human if all the safety checks are in place and everything like that. Right? So cell in a dish. That's usually where it all gets started. So you can buy it from online. Like I don't think you even need like a special license to buy it. Like anybody can buy it, and they're not cheap. Okay, 20 microliter, a microliter. That's same as micrometer, right? A liter has 1,000,000 microliter. So this is very, very, very small amount, and it costs $486. Okay? And it's understandable because once you buy it, you can grow your own supply, right? Yeah, like I've actually done it in my graduate work. You can grow these cells, and then you can separate them and freeze them in little tubes, put them in minus 80 degrees. And when you need new ones, you just store one and then you grow more. Okay, so that's why they're so expensive, because usually people don't go buy brand new ones unless you kill off your whole stock. So there was actually like a lawsuit against the company that is selling the HeLa cells. The company is called Thermo Fisher. The estate, the lady died, right, from the cancer, but her family sued the company because they said they didn't actually get her consent for profiting from her cells, right, and propagating it without her consent and selling it without her consent. And so the company tried to countersue them, but I think ultimately the estate wants some money, and the company actually pay for the damage that they have caused to the family. You can search more about this if you're interested. So we were talking about the 4 parts of the cell theory, right? Living things are made up of 1 or more cells. Number 2, each cell is individually alive, even though they're part of a living organism. And the next part is of the cell theory is that all living cells come from division of pre-existing cells. Right? To get more cells, you have to start with a cell. Right? The cell would split into 2. In fact, here is 1 cell. And when it divides, you will create 2 identical cells. This process has a name. It's called mitosis. We will learn more about mitosis later on in the course. Right? But to get more cells, you need to wait for the cell to divide. Okay? Cells also, this is the 4th part now, cells also contain hereditary material. Okay, so these are like DNA, generally speaking, which are passed on during division. Okay, so last time I asked you guys, right, like how many chromosomes do you have? Does anybody remember? How many chromosomes do you have in your cells?
46.
Mm-hmm. Yep, so you start with 46 chromosome. And when the cell divides, you actually need to copy all your DNA. And in the end, each cell will have 46 chromosome. All right. In the picture here, you are actually looking at onion cells. Again, the color is not the natural color. We often stain them to highlight certain features. And you know, this is an onion cell, and this actually is an onion cell that's dividing, in the process of dividing. All these things that you're looking at are actually chromosomes. Yes, Fahima, go ahead.
When we divided it, then it's 23, 23. It should be like that.
No, because you actually make a copy of all your chromosomes before you divide. Okay, so 46, you actually double the amount before you divide. So then, in the end, each cell is still going to have
23,
46.
23.
No,
Oh,
46, 46.
sorry,
So
sorry.
yeah, because you double it first, right? I didn't write
Okay.
it here, but you actually have to copy all the material first. Does it make sense?
Yeah,
Yeah.
like that.
Okay.
Yeah.
Good, good, good. Okay, so to study cells, we have to use a microscope. The first microscope was actually created by this Dutch scientist. He actually wasn't a scientist before. He's like I think he was like a tailor. It's like selling clothing for people. But he had a side interest in creating these lenses. Okay, so one day he created this small lens, and he was looking through it, and on the other side was some water, and he found out that there was something moving in the water, and he actually described them as I'm just gonna write animalcule, something like that. Okay, animalcule. Okay? It's like molecules that look like animal, because they're like moving around. And he got really fascinated. So he kept on refining his design of the lens, and eventually he created his first handheld microscope that looks something like that. And you know, he actually made very detailed record of everything he saw under his microscope. But people didn't believe it. People thought he was just imagining it, because no one was able to recreate the result. Okay, no one was able to create the lens. They didn't have the skill that it took to create the lens, and so people thought he was just making this stuff up. So he actually invited people, prominent people in the society, I guess, at the time, and invite them to look through his instrument. And you know, he had lawyers and people who vouched for him that, you know, we actually saw what he was saying, right? And you know, that started the whole field of like microscopy. Nowadays, microscopes are much more sophisticated than the handheld one. This is a typical light microscope. Okay, light microscope. If we were having this class in person, then you would be able to use the microscope and you know, actually look at specimen. But this being an online course, of course, we won't be able to do that. But light microscope are not too expensive. Okay, a couple $100, you're going to be able to get a pretty decent one, and you are able to magnify things usually up to 1,000 times. There are much more powerful microscopes that are being used. These are typically reserved for research or being used in a hospital setting for certain kind of diagnosis. And there are two kinds here. This one is a TEM, and this one is SEM. Never mind what they stand for, okay? The key thing here is the SEM gives you a 3D view of the specimen. Okay, and here the TEM gives you like a cross section of your specimen, right? So cross section is good for certain application, and 3D view is, you know, also good for certain application. If you want to look at the inside of these things, then you need the TEM. But if you want to get a overall general view of what it looks like, then you know, you got to use SEM. And this line here is 1 micrometer. Okay, so these structures are really small. Just curious, anybody know what this might be? It's somewhere in the body. You have it, I have it, everybody has it. Do you know what might that be?
The internal layer for intestine.
Okay, that's a very good guess. Yeah, I get that a lot. Okay, but not quite though. Anyone else?
Maybe for respiratory system?
Yes. Okay, so this is in the airway. Okay. In your airway, you have these little hairs that will help you sweep up like dust that you've inhaled, and bring it back up to the nose or to the throat, right? So you can get rid of it. Okay, so that's like these microscopes are really big, and they are like super expensive. Yeah, if you order the whole set, it's easily go up to 2.5. Sorry, they can magnify up to 2.5 million times, and the costs are easily in the millions of dollars. Okay, compared to the previous one, the light microscope is just a couple $100s, right? So these are not your typical ones that you will be able to afford at a school, right? Maybe university. All right. Let's see if you are able to find the right answer here. Okay, over here, organelles are the basic unit of life. That's not true, okay? Cells are. Simple cells can arise spontaneous from rotting vegetation. No, that's not true. All cells come from the division of pre-existing cells. Only complex organisms are composed of cells. Nope, all living things are composed of cells. This one is the right answer, okay? A cell is the basic structural and functional unit of living things. Right? So we are all made up of cell. That's what basic structural unit means, and they function in a way that help us function as an organism. Okay, this is another one. Okay, good. Right. These are the common things, and this is the thing that is the exception. Okay. We haven't even learned about what a nucleus is yet. Okay, we will in a second. Okay, next we're going to talk about the cell membrane and the nucleus. So, when it comes to classifying cells, we have two categories. Okay? We have what's called eukaryotic cells, and sometimes you can just say eukaryotes. Okay? Eukaryotic cells or eukaryotes. Now, these cells are typically more complex and they are bigger than the other type of cells, which are the prokaryotic cells. Right? And the main feature that separates eukaryotic cells and prokaryotic cells is the presence of a nucleus. Okay, so there's usually like a circular structure that is in the middle, and that is where you will house all your genetic information. We'll write all these down later. The eukaryotic cells have many types of organelles with specialized functions. Animal cells and plant cells are examples of eukaryotic cells. Okay, the amoeba and the paramecium, they are also eukaryotic cells, but actually these are not like animal cells or plant cells. Okay? There are other things besides animal and plants. Okay? You should know that. Okay? They're fungus, for example. There's something called protists. So these are not animals or plant cells, but nevertheless, they are examples of eukaryotic cells as well. Okay, so all these things that you see in the picture, the mitochondria, ribosome, Golgi body, all these are organelles. Okay, organelles are structures inside a cell with specialized functions. Okay. On the other hand, we have the prokaryotic cells or prokaryotes. Prokaryotes, okay? They P stands for primitive. Okay? They are very simple compared to eukaryotic cells. In fact, at the beginning of like the early lives on Earth, began with prokaryotes, okay? And later on, the eukaryotic cells came. The prokaryotic cells are smaller, simpler in structures, and the defining feature that separates them from eukaryotic cells is that they do not have nucleus. They don't have a circular structure to contain its genetic material. Its genetic material is just kind of like lying around on its own. There are only a few type of organelles. Most bacteria are prokaryotes. Does anybody know any bacteria off the top of their head? Like, yeah, bacteria name that you've heard on the news, maybe you read about.
Like Salmonella?
Yeah, sure, right? Salmonella is a bacteria. Anything else? Like, a lot of time they have recall.
E. coli.
E. coli, yeah. Okay, so Salmonella, E. coli are common examples of I think I spelled the Salmonella wrong, so it's
So something like a Streptococcus and
yeah, Streptococcus, it gives you strep throat, right? Yeah, so all these are examples of prokaryotic cells. Okay. All right. Regardless whether it's eukaryotic or prokaryotic cells, there is going to be a cell membrane that surrounds the cell. We learned from last week that the cell membrane is made up of primarily these molecules called phospholipid, right? Phospholipid. And there are actually two layers of phospholipid. The membrane is selectively permeable. Selectively permeable means it lets some things pass through while blocking others. Okay. So if something is like completely permeable, then anything can go through. If something is non-permeable, nothing goes through. If it's selectively permeable, then it chooses what can pass through. And the cell membrane is a barrier that separates the internal environment, which we know is called the cytoplasm, from the external one, okay? The extracellular fluid. Okay. So the phospholipid bilayer makes up the cell membrane. We know that already. And just to recall, the phospholipid actually has two parts to it, right? There is a hydrophilic head, which is made up of the phosphate group. And then there are the hydrophobic tails, which are made up of the fatty acids. Last time I told you, you can think of the cell membrane as a peanut butter sandwich. Yeah, the bread forms the heads, and then the peanut butter in the middle forms the hydrophobic core. And if you use a TEM, the microscope that I showed you that can magnify many times, you can see this here is actually the cell membrane. Okay, so like if you zoom in onto the square and you draw it out, it basically looks like this. Okay, the white line in the middle, that's where the hydrophobic tails are. And the two dark black lines, that's where the hydrophilics are, hydrophilic heads are. Okay. Now, the cell membrane is not the only place you have these phospholipid bilayer. Some organelles, as you will see, that are inside the cell, they have their own membrane as well. Okay? And those membranes are also made up of phospholipids. Okay, so things can get a little bit abstract for the next little bit. I usually like to draw this out with the class. I mean, it depends what kind of person you are, okay? But generally, people tell me drawing it out with me together helps them understand it a little bit better, okay? So like if you're not like not into drawing or something, then maybe you can just sit back and watch, and then you can decide to draw it out or not later on. But give it a try. Hopefully, drawing it out is going to be a little bit easier for you in terms of understanding what's going on. So get a new piece of paper like this. We're gonna start. We're gonna use a whole piece of paper. Okay, we're gonna start here, and then we're gonna start